Hi, I'm Andrew, and this is the Peptide Bracelet Kit activity tutorial video. I'll be going over the steps of how to make your very own bracelet. So don't worry if your kit looks a bit different than this. We have a variety of different packagings, but they all contain the same supplies. Also, each kit will probably have a different color of beads, so don't worry about that either. So inside the kit, we have 3 pieces of paper, which are your instruction sheets. I'll go over those really soon. And we will have at least 12 beads of each color. There will be 4 colors. And then we'll have a piece of string or yarn for the bracelet. The first page of the packet is the introduction page. It has a paragraph of information that you should probably read. And there's also 3 QR codes here that you can scan, and you should watch those videos before beginning the activity because they will give you the background knowledge you need to succeed. In this activity, we will be creating the messenger RNA template sequence that could basically create the cochinein peptide. A peptide is a very short protein, and in this case, cochinein is made by a type of bean. That's right, like a bean. And they have shown antifungal effects based on lab experiments. So basically, scientists have found that these peptides can kill fungi. They've also been shown to hurt leukemia cells, which is a type of cancer, and additionally inhibit viral enzymes in HIV. So they're a pretty powerful, potent little peptide. So once again, make sure to watch these 3 videos before getting started. On the second page, there'll be this really big, bold sequence of letters. This is the cochinein amino acid sequence. If you don't know what an amino acid sequence is yet, don't worry, it will be covered in the videos on the previous page. So make sure to watch those first. What we'll be doing in this activity is to look at each letter of the amino acid sequence, each letter being an amino acid, and we'll figure out which messenger RNA nucleotides could or would represent each of the amino acids. Here, there's a little table where you can write in which color you'll use for each bead. For instance, in this example, I will use the color yellow to represent adenine, and I will use the color pink to represent uracil. You can pick whatever combination of colors you want. It is helpful to write it down into the table provided so you can keep track of what's going on. So what did I say? Adenine is yellow, uracil is pink, cytosine will be red, and guanine will be green. Adenine, uracil, cytosine, and guanine are the 4 RNA nucleotides. They are like the building blocks of all RNA molecules. If you don't know what they are, make sure to watch the videos I previously mentioned, and they'll cover what they are. Okay, so the final page of the packet is probably one of the most important ones. It has a codon chart and a table of amino acid names. So a codon chart is a really useful chart that tells you which messenger RNA codons, which are 3 nucleotides at a time, create what amino acid. For example, GUU, GU, is a messenger RNA codon that creates or encodes for VAL, valine. Meanwhile, GAG, GAG, can create GLU, which is, I believe it is glutamine. Nope, sorry, glutamic acid. Note that some of these amino acids can be created by more than one mRNA codon. For example, LEU, leucine, has 4 different ways to spell it: CUU, CUC, CUA, and CUG. Whenever there's more than one possibility, it is up to you to pick which one to use. So, before we get started, there's one last thing. There's a bioinformatics extension activity, which is completely optional. It might be better if you're interested in coding, and it will be going over how to use Python, a popular programming language, to convert DNA or RNA to protein sequences automatically with a computer. Okay, so let's get started with a demonstration of activity. I'm going to get out my string. So the first amino acid we have is K. So, just seeing the letter K, you don't really know what that means, right? Like what amino acid is that? So we're going to go to our handy table on this side, and we're going to look for which one is K. So as you can see, K is lysine, Lysine, which is abbreviated as LYS. So that means the first amino acid in the cotonin peptide sequence is lysine, also known as K or LYS. Now, let's go over to our handy dandy codon chart and find LYS. There it is. And you can see LYS can be spelled by 3 A's or an AAG in terms of messenger RNA nucleotides. And remember that each group of 3 nucleotides is called a codon. So in this example, I'm going to go with AAG to code for lysine instead of AAA. No real reason. Maybe I just want to put on a green bead. So AAG. Remember that I wrote down adenine A as yellow and guanine G as green. So I'm going to stick to that here and put on AAG. When you put on the first bead, you want to be really careful and tie it with a double knot or triple knot so it doesn't come off at the end. And then you'll be left, and then your bracelet beads will literally all fall off and you'll have to start all over. And just be careful because the string can be pretty bendy and loose. I would probably recommend triple knotting it just to make sure. In total, the bracelet will have 30 beads because there are 10 amino acids in the cotonin peptide sequence. And remember that each amino acid is coded for or represented by 3 messenger RNA nucleotides, and 10 times 3 is 30. Okay, so it's still not being that tight. I'm just gonna pull it together one more time. If your kit has this little stretchy plasticky material, you'll probably want to tie it a bunch of times to be safe. But if you have yarn, it should probably be better at tying. Okay, so AAG, I put on one yellow and I need another yellow and a green. Make sure to put them on in the correct order because order does matter. Otherwise, you could be creating a different amino acid. Here we go. AAG. Okay, and I'm going to do the next one as well, just to get us started. And we have a Q. So what does Q stand for? Again, back to our handy dandy table. Q is GLN, also known as glutamine. And it's funny that it's spelled with Q. So remember, you cannot trust the one letter codes because it's not always the first letter that starts with, like E stands for glutamic acid, which definitely doesn't start with an E. Okay, so going back to our codon chart, glutamine, GLN, or Q, can be coded by CAA or CAG. Let's go with CAG. So CAG would be a red, a yellow, and a green. Okay, awesome. And we're already 2 amino acids down and 8 more to go. At the end, make sure to securely fasten and tie up the string to make sure your bracelet doesn't come apart. Again, you may want to tie a lot of knots. one thing to note is that in the packet, there's a link to, it says helixbio.org/peptides. This is a website where we will be uploading a list of other resources and any corrections that may be needed to be made in case we made a mistake in the packet. So make sure to check that if you're confused about something doesn't make sense. So overall, thank you so much for getting your peptide bracelet kit. And I really look forward to seeing what you create, and I hope you learn from it. When you finish your bracelet, if you want to, and if you have parent permission, send us a DM on Instagram at the Helix Initiative with a picture of your bracelet, and we would love to share it on our platforms. Thank you and happy learning.